So Dr. D'Arcy, as I mentioned, completed her Bachelor of Science at the University of Sydney in Australia, and she completed her PhD in biochemistry at the University of Cambridge, where she utilized X-ray crystallography, and then she went on to Dr. Carolyn Luger's lab at Colorado State when she was still there and did her postdoc there. And I actually remember when I was a graduate student hearing a lot about the hydrogen-deuterium exchange work that Sheena did, and I was like super like geeked out about it. I thought it was so cool. So I'm really excited to hear Dr. D'Arcy present today. So as I mentioned, Dr. D'Arcy is at the University of Texas Southwestern in Dallas, where she started in 2015, and her lab has grown. She has 10 graduate students, which is really cool. I'm really excited to hear what you all have been working on now. And I will, I guess for the sake of time, I'm going to stop talking and I will give the floor to Sheena. Thank you so much for being here.
Great, thank you for having me. Okay, so before starting, I actually, in the spirit of Postdoc Appreciation Week, I want to give a very specific shout out to all of the postdocs that are in the audience and also to the organizers. I'm not sure if everybody is aware, but I believe the majority of the organizers are postdocs. So yeah, we really appreciate you postdocs. But today, I'm going to present some work that my lab has been doing in developing and implementing a solution assay for measuring the dynamics of various different chromatin complexes. Now, this image here is not an image that I need to introduce to this audience. It is, of course, the crystal structure of the nucleosome, which is the basic building block of chromatin. What I do want to do, however, is point out some details that the non-structural biologists in the audience might not be aware of, as I will be referring to these details throughout my talk. The first is that the 4 core histones share a central core fold, and that this fold is composed of a short alpha-1, shown here in H3, a long alpha-2, and then a short alpha-3. And this core central fold is responsible for many of the histone-histone contacts we see in the nucleosome. So these histone-histone contacts are primarily these 4 helix bundles, such as we see here between the 2 copies of H3, as well as a 4 helix bundle down here between H2b and H4. Now, other histone-histone contacts that are really important to the nucleosome involve this C-terminal region of H2A, which nestles here amongst the H3-H4. Finally, in the nucleosome, we don't just have histone-histone contacts, but we have Histone-DNA contacts. And these are mediated primarily by these loops, these DNA binding loops on either end of the histones. So you can see these are on the surface of the histone octamer, and they're responsible for wrapping the DNA around the octameric core. Now, the structure of nucleosome has been known for quite a long time, and what we also know is that this structure is regulated, and it's really important to regulate this structure and its interactions with other nucleosomes in the cell. There are various factors that are important to the regulation, some of which are listed here. Now, these factors act alone and together to really balance this equilibrium between nucleosome assembly and disassembly. Now, in some cases, this equilibrium is known to be balanced by an alteration in nucleosome structure or stability. The precise mechanistic underlyings of these regulatory factors, however, are not really known. We have many static structures available, and they're absolutely invaluable, but they often have very subtle differences, or in many cases, there are structures that are unavailable, enabling us to really question how these different factors influence chromatin structure. Now, in response to this, my lab set a goal, which was to develop an assay for the quantification of chromatin structure and dynamics. Now, we wanted to use this assay to look at how these regulatory factors and combinations of these regulatory factors affect the nucleosome, whether that be at the histone level, the free histone level, at the level of a nucleosome intermediate, the nucleosome itself, potentially a multinucleosome array, and even looking at multinucleosome arrays with different binding partners. Now, we had several criteria that we had for this assay. We wanted it to be in vitro, at least to start. We wanted it to be in solution, amenable to a high throughput format, and of course, in keeping with this, it needed to require low sample consumption. And most importantly, we wanted it to be quantitative. So when we look at a static structure of a nucleosome or a modified nucleosome, it can be difficult for us to know how the different interfaces that we see between the histones, as well as between the histones and DNA, how they actually contribute to nucleosome stability. So in response to this, we decided to use a setup that made use of comparative hydrogen-deuterium exchange coupled to mass spectrometry, or HDX. Now, what HDX does is it measures how many hydrogens in the protein backbone are able to exchange with deuterium from the solvent. So we can take our histones in various conditions, so condition A versus condition B, and we can expose them to deuterium. Now, at different time points, we will stop the exchange reaction and we will quench the reaction and denature the protein. Now, the reason we want to denature the protein is that then we chop it up into lots of little peptides and we measure how much deuterium is in each of those peptides. This enables us to localize which of the protein backbone amides are involved or have altered hydrogen bonding or solvent accessibility. So we're essentially measuring the rate at which the deuterium is incorporated. We really care about this rate, like I said, because it's influenced by hydrogen bonding and solvent accessibility. And these are things that are altered by conformational changes of a protein or when the histones form different interactions. Another key reason why we wanted to use hydrogen-deuterium exchange is that we could really make it quantitative. In other words, the strength or frequency of which an interaction is formed is reflected in the amount of deuterium that's incorporated. So between condition A and condition B, we would be able to say this interaction plays a more important role or a less important role. Now, when you do an HDX experiment, you get a lot of data. Right? We chop these proteins up into peptides, and we often have several hundred peptides. For each peptide in each condition, we'll have a series of spectra that may look something like this. So this is our spectra of our peptide at T equals 0, so when we have no deuterium incorporated. And then over time, you can see that this spectra is shifting to the right. And we can calculate how much deuterium is incorporated by looking at the difference between the centroid here compared to the centroid here. And we can graph this on this uptake plot where on our x-axis we have our time exposed to deuterium, and our y-axis we have the amount of deuterium that's incorporated. So we will get a plot like this for each peptide. Like I said, we have several hundred of them, where each trace here represents a different condition. So we certainly need a way to collapse the data so that we can sort of look at it all at once. So in today's talk, what I will be focusing on is the difference in deuterium uptake in different conditions. And I will, most of the time, project this on this heat map, where on our x-axis here we have our peptide boundaries, our y-axis we have our time, and the intensity of the red indicates the difference in D uptake between two different conditions. So anything that is dark red means the two conditions that we're comparing have a very big difference in deuterium uptake. In other words, a difference in hydrogen bonding or solvent accessibility for that part of the protein. So today I'm going to present the data that we've collected for free histones, for the histone Octamer, the nucleosome, as well as the tetrasome. And when we look at these data as a whole, we really are able to quantify and reinforce findings that we already know from years and years of studying the nucleosome. And so this really validates the use of this approach. And we also have a few new findings that we've found out along the way. So the first comparison that we did was we wanted to look at free histones at different salt concentrations. And we were motivated to do this, as anybody that works with chromatin in vitro knows, we really use a decrease in salt concentration to promote nucleosome assembly. And traditionally, this is thought of as regulating the interactions between the histones and the DNA. But when we looked at our histones, our free histones, our histone heterodimers at different salts, we got a very surprising result. And what we saw was that as we increased our salt concentration, we saw a lot less deuterium uptake. So here I am sort of incorporating all of the data together, where each of these rectangles represents a different peptide, and we're doing the cumulative difference between the two extremes of 0.2 and 2 molar. And what you can see is that for the histone core, the alpha 1, alpha 2, and alpha 3, as we increase the salt, we're really seeing a dramatic increase in how stable the histone fold is. So we see this in H2A, we see this in H2B, we see this in H3, and of course, as I'm sure you're all about to predict, we also see this in H4. Now, this was really surprising, and you know, when we line up these heat maps here according to where the cores are, what you can see is that we get very similar result for all of the histones, with the greatest degree of stabilization occurring at the center of alpha 2. So that center of alpha 2 is right here where the two monomeric chains in the histone heterodimers will cross. Now, when we did this, we started to wonder, you know, is this something that's unique to histones, or is this something that we would see in any protein? In other words, if we increase salt concentration, do we expect to increase hydrogen bonding? So in order to test this, we turned to other proteins in the lab. We said, do they show the same salt-dependent stabilization? To do this, we used linker histone H1, and we chose it because it has a similar PI to the histones, a similar secondary structure, but it's monomeric. So we were really asking if it was the heterodimer of the histones that was causing this salt-dependent stabilization. We also used Apo-Myoglobin, where here the variable we were testing was really PI, because it has a much more neutral PI than both the core histones and linker histone H1. So these are the data for linker histone H1. What you can see is, again, we see this salt-dependent stabilization. But finally, with myoglobin, we did not see this salt-dependent stabilization. You can see we have almost no change here as we go from 0.2 to 2 molar salt. The myoglobin, however, did give us a hint as to the mechanism of why this salt-dependent stabilization was happening. And that's because this very, very small change we see at 2 molar salt in these two regions of the protein showed us that this was where the lysine residues were located. So there's this double lysine residue, 62, 63, and these triple lysine residues from 77 to 79, really suggesting that this salt-dependent stabilization is most likely attributed to the histone basic PI, and is potentially something that could be modulated by neutralizing these charged residues, potentially by acetylation. Another interesting observation that we got in the free histone data is that the histones were cooperatively unfolding at their dimer contact points. So I'm showing in yellow and red over here, and in blue and green over here, the regions of the histones for which we detected cooperative unfolding. And by cooperative unfolding, what I mean is that all of the hydrogen bonds in this region of the protein were being broken and incorporating deuterium before they were being made again in a cooperative fashion, so all at the same time. So this really told us that the histones, these heterodimers, are breathing. You know, they're not completely coming apart, but at their points of contact, they're definitely breathing with regions of the histones unfolding. And we were able to detect this by this bimodal distribution that you can see in some of these spectra. So this would be the exchange of the unfolded population versus the folded population. Now, as we increased our salt concentration, this was dramatically reduced. So we really think that that in vitro nucleosome assembly is motivated not only by controlling the histone-DNA contacts, but also by stabilizing the structure of the histone heterodimer. So from our free histones, we can conclude that hydrogen bonds in the histone fold are stabilized by salts, and that this likely contributes to in vitro assembly. The salt stabilization is not a general phenomenon of all proteins and likely relates to protein pi, and that the histones cooperatively unfold at dimer contact points. Now, I haven't shown the data today, but my lab is also very interested in histone chaperones. And what we've shown is that this salt stabilization in fact mimics the activity of histone chaperones. And so perhaps the need for the histones to be stabilized has sort of led to the role of histone chaperones in the cell. So moving on, we next asked about the dynamics of the histones in the octamer. And here the key was, can we identify and rank the histone interfaces? Can we say which ones are more important to octamer stability? When we first did this, these are the data that we got. And so in these heat maps here, what I'm showing is the difference between the free histones and the histones in the octamer. And my student brought me these data and he said, oh, there's no changes. But of course, you can see there are some small changes here. And we were really encouraged because when you look at where these changes occur, right, we know that these are really important regions that are used in histone-histone interactions in the octamer. For example, this is the C-terminal tail of H2A that I pointed out nestles in amongst the H3 and the H4. And this is the H3 tetrameric interface. But we obviously wanted to see if we could enhance these interactions and maybe detect some of the ones that we didn't detect here. So we thought rather than being in this, what we now classify as a highly stringent condition, we tried to do the exchange under low stringency. And so the way we did this was we reduced the temperature to 4 degrees because that's known to increase the stability of the octamer. And we also did it at 5 times the protein concentration. And what you can see is that all of these very small changes that we saw are now enhanced in our low stringency condition. And we're also able to detect some of these other interactions. Now, it's very difficult to sort of imagine what interactions we're detecting them in this type of presentation, but we can of course show the data projected onto the octameric structure. So again, the colors I'm using here reflect the difference in deuterium uptake between the free histones and the octamer. And what you can see is that these changes are absolutely localized to the histone-histone interactions that we see in the nucleosome. You know, I'm particularly impressed that we're even able to detect this interaction on the underside of the octamer between the 2 copies of H2A here. But we really wanted to sort of benchmark our approach. So what we did was a PISA analysis, and we compared our HDX data to the PISA analysis. So PISA analysis is when you submit the crystal structure online, and it will give you a really nice breakdown of all of the interactions that are seen between chains in the structure, as well as giving you a surface area. So these are the major interfaces that we detect, that are seen in the crystal structure of the nucleosome, and this is their surface area, and this is whether or not we're able to detect them in the HDX. Now, the take-home message here is that our degree of protection pretty much correlates closely with the surface area of the interaction. And the surface area of the interaction is one way to sort of quantify its importance. However, we do have one exception here, and the exception is the 4-helix bundle between H2B and H4. So this is interface 3 shown here. So we do not detect this at all in our high stringency, and we detect it only a little bit in our low stringency, which of course would not be predicted based on its surface area. So what this tells us is that in the octamer, not in the nucleosome, but in the octamer, that this is a transient interface. In other words, the octamer stability is really driven by this C-terminal tail of H2A interacting with H3H4 here, with this interface constantly being broken. So you can imagine the H2A, H2B kind of swinging out, just being held on by the H2A C-terminal tail. And I say that these data are for the Octamer, but we actually see a similar result, as you'll see in a few minutes, in the nucleosome. So the stability of this interface is lower than we would predict based on its surface area, even when the DNA is present. As for the cooperative unfolding, we still see it in the Octamer. However, the regions that cooperatively unfold are different. We're no longer seeing it at the dimer contact points, but rather we're seeing it at the primary interfaces between the histones. So in the context of the Octamer that's stabilized by salt, these interfaces are clearly being broken and reformed, albeit not all at the same time. So from our histone Octamer data, we can clearly show that Octamer formation is driven by the H2A C-terminal docking domain, as well as the H3H3 4-helix bundle, with the H2BH4 4-helix bundle appearing less important. And we see cooperative unfolding at the major interfaces between the histones. Now, I think this weakness of the H2BH4 4-helix bundle is really important because this is a vulnerable interface that may easily be exploited by histone chaperones and other regulators. Now, finally, the piece de resistance is, of course, our HDX experiment with the nucleosome. So what I'm showing here are a variety of comparisons. The top two is just a repeat of the free histones versus the Octamer. We always do this sort of as a control at the same time so that we can quantify our comparisons. But the one that's of most interest is the comparison in the middle, which is our Octamer versus our nucleosome. So the goal here was really to see if we could detect those Histone-DNA interfaces. Down the bottom here, I'm showing the data I've already presented for the stabilized Octamer that was done at high concentration. And it's really good to compare because you can see that the histone-histone interfaces we detected here are absolutely seen here when we compare our Octamer to our nucleosome. So it's telling us that the DNA and the salt are having a similar effect on the histone-histone interactions. But we're more interested in what's new, such as, for example, this extra protection we now see here at this loop between alpha 1 and alpha 2 that we did not see down here. This suggests that this is a histone-DNA interface. So these are the data for H2H2B. These are for H3H4, but of course it's most easily interpreted if we project it onto the structure. And what we can see here is that the HDX clearly identifies the trajectory of the DNA on the histone Octamer, with the protection being localized to the surface of the Octamer that is known, of course, to wrap the DNA. But we have a few interesting observations. So like I said, we see an enhancement of the histone-histone interaction, which is proportional to that we see with 2 molar salt and high concentration in the free histone Octamer. We no longer detect cooperative unfolding of the histone-histone interfaces, but we do detect cooperative unfolding of this alpha N helix here of H3. Now, this is really interesting because this helix bridges the DNA as both strands of the DNA as it wraps around 1.6 times, suggesting that this end of the DNA is definitely visiting an open conformation. I guess one of the things I was most excited about in terms of it being quantitative is that the amount of change we see in the DNA binding lips of H2B depends on their proximity to the end of the DNA. In other words, we see less protection at the ones at this end compared to those at this end. Again, this really shows us that we're able to detect this opening of the DNA, which of course we know from various other biophysical approaches. Now, to really test if we can detect these sorts of dynamics, we went on to look at the tetrasome. So the tetrasome is the H3H4 with the DNA, so there is no H2A, H2B present. So here I'm showing a comparison between the free H3H4 compared to in the tetrasome. You can see that most of the differences are situated at the dyad of the DNA. So that's what's positioned right in the middle of the H3 4 helix bundle, with the changes being reduced as we move away from that dyad. This is our exception here because this is a DNA binding loop in H3. But one really interesting result here is that we see no changes in the alpha N, which I'd previously shown interacts with DNA in the nucleosome. This tells us that this alpha N is not involved in DNA binding in the tetrasome. It in fact has a conformation identical in free histones as it does in the tetrasome. So now if we look at the difference between the tetrasome and the nucleosome, you can see that we almost get the inverse data, right? So where we had low here, now we get more. Low here, now we get more. So here we get more differences as we move outward from the dyad as that DNA is being wrapped. Now we see this stabilization of this alpha N as well as this alpha 3 of H4, but everything makes so much more sense when it's projected on the structure. So on the left here, I'm showing the changes we see as we go from free H3, H4 to tetrasome. You can see they're really centered around the dyad with these two regions not at all being involved or weakly being involved in DNA binding, with the alpha N here not being involved at all. Compared to when we go from the tetrasome to the nucleosome, now we're seeing protection here as well as here. Now, these data are really consistent with the molecular dynamics simulation that was done several years ago that shows in the tetrasome, these regions of H3, H4 are really visiting an unfolded conformation. So in conclusion, we can clearly use HDX to quantitatively measure histone unfolding, histone, and Histone-DNA interfaces in various chromatin substrates. So now we're frantically using the system to characterize the effect of various regulatory factors. We'll skip over to this and make sure I have time to acknowledge the students that did all the work. This was spearheaded by a graduate student in my lab, Darby Ball. He's absolutely phenomenal at doing these, very highly analytical, precise experiments. And he was assisted by an undergraduate, Adam Timmerman. And of course, I would like to thank our funding sources. This is funded by an R35 from NIGMS, ASBMB, the UT system, as well as startup funds from UT Dallas. And with that, I'll take any questions.
Thank you so much. That was fantastic. I'm always excited to see this approach. I always kind of am curious, have you looked at any weird histones, or weird complexes like overlapping dinucleosomes or modified histones or any of the oncoproteins or oncohistones? I feel like
Yeah.
the possibilities are endless with this. I think it's super cool.
Yeah, so that's the goal, right? So when we started, I will share. I think a reviewer in a grant said it was like Hershey's heaven, because now we have the assay and we can know exactly what interactions we're detecting when we see change. Now we can do it high throughput and really start to test these things in a combinatorial fashion. So I think the first thing we're going to do is probably look at some of the histone variants, because all of the crystal structures of these histone variants really show almost an identical structure to the canonical nucleosome. So that work is ongoing. And then in terms of the dinucleosomes, so that's phase 2. I did have a slide here.
Oh, cool. That's exciting.
So our goal here is to move to array systems where we can actually tell the difference between nucleosome 1, 2, and 3. And so the way we're going to do this is to use these isotopically labeled nucleosomes.
Awesome. I will mention Siavash Kurdistani also asked about histone variants too, so I guess he's probably excited to hear that you're working on that. So really cool. And then we also have a question from Greg Bowman. He says, great talk, I agree. I was wondering if you could comment on how salt is stabilizing the positive charged region similar to DNA. Does that mean we should be thinking differently about DNA stabilization?
Yes. So if you're talking about the salt stabilization for the free histones, actually, I think this is probably a general phenomenon for all proteins that have, you know, highly charged residues, sort of localized, and it's the idea that the charges there are really constrained by the salt, and in this case, it's kind of springing the histones to unfold. In terms of the role of salt in stabilizing the octamer, I think it is really just acting as a mimic of the DNA. And the reason I say that is because the interfaces we see and the degree of change that we see is really similar to when we actually have the DNA there in the nucleosome. Although the DNA, of course, does a much better job because I guess it kind of kinetically traps them into that octameric conformation.
Another question from Siavash Kurdistani. He asks, why do changes in concentration of the octamer affect the degree of exchange? Aren't these intramolecular interactions?
Yeah, so the reason that the concentration affects the degree of exchange is because it's basically increasing the percentage of the histones that are actually in that octameric conformation. So while a lot of chromatin experiments are done at really high concentration and people say, oh, the octamer doesn't fall apart at 2 molar salt, one of the advantages of HDX is that we do these experiments at low concentration. So during the exchange, when we had it really low, it was just falling apart in keeping with the affinities of those interactions in the absence of DNA.
I think we're actually exactly on time. So I think we will go ahead and wrap up the seminar today.